Channel 2 News. My name is Sydney McCracken, and I'm going to be hosting our very next segment. Something that I did not know existed, yet totally does. Today, we are going to be talking about the genetics in racehorses. I would like to introduce you to my three special guests. We have Miss Bianca. Ladies, thank you so much for joining us. We're going to go ahead and start with Bianca today.
When we think about the genes involved in the neurological aspect of these thoroughbreds that are selected for racing, we have three important ones: KANA1, NTM, and SYNDEG1. When we think about the KANA1, we think about motor functions, execution, movement. Those are very important aspects when we think about an athlete as a horse. It's the same to think of a human that has the ability to quickly learn how to move the feet very quick, how to stay in equilibrium, like we close the eyes, and how long can you stay standing that. So that would be related to KANA1. And if we look at that, then we can find it a very good connection in neurological with run. And if we look at where these meet KTN1, the first one related to motor and execution in humans, we can find this is the brain on the forebrain, which is usually the top section over here, because then we have the hindbrain and the middle or midbrain over here. But yeah, these will usually be found similarly genes in humans will be over.
Well, thank you very much, Bianca. That is some very exciting information. Next up, we are going to hear from our muscle and skeletal muscle specialist, Jillian. Take it away.
I will cut this out. The biggest two that we will talk about determine the muscle phenotype in racehorses, especially those that influence the skeletal muscle. That will be the HDAC9 gene and the MYLK2 gene. The skeletal muscle is a highly adaptive tissue that responds to exercise and training by increasing muscle mass by changing fiber type composition with mitochondrial functional adaptations. The HDAC9 associates with maximal oxygen uptake response to training and encodes protein that inhibits skeletal myogenesis, which is involved in cardiac muscle development, as well as regulating muscle growth. Thoroughbreds were found to have the highest frequency of HDAC9 gene, and racing breeds were found to have 3.4 times more frequency of the HDAC9 gene than non-racing breeds. The MYLK2 gene encodes myosin light chain kinase in skeletal muscle. It plays a critical role in muscle contraction and functions in skeletal muscle satellite cell differentiation, which are responsible for postnatal growth, regeneration, and hypertrophy. It also functions for regulation of muscle filament sliding, which plays a part in contraction of muscles. In humans, the MYLK variants are associated with phenotypic response to exercise-induced muscle damage. This held a significant difference in racing versus non-racing breeds. The highest frequency was found in Arabians, and the lowest were found in breeds like quarter horses and standardbreds. Metabolic properties of the skeletal muscle influenced by slow and fast type muscle fibers defined by myosin and are characterized by the difference in densities and functional properties of the mitochondria. It ensured adequate supply of ATP to meet energy needs required of the skeletal muscle using glycolysis and oxygen. Glycolytic requirements for high-intensity exercise are likely responsible for the observed varieties among breeds.
Well, folks, you heard it here. Racehorses have a core set of genes that have helped them excel in multiple different ways in the racing industry. These genes affect neurobiology, muscle, and even metabolism within the muscle.
The horse breeds that have been discovered to have the speed gene for racing are the thoroughbred, the Arabian, and the Mongolian racehorses. The thoroughbred originates from 3 founding sires, the Darley Arabian, the Godolphin Arabian, and the Byerly Turk. They were bred for racing and jumping, which the breed excels at to this day. The Arabian originates from the Bedouin people around 2500 BC, and they were originally bred as a war horse on the Arabian Peninsula, which is currently the location of Bahrain, Kuwait, Oman, Saudi Arabia, the United Arab Emirates, and Yemen. In contrast, Mongolian racehorse originates from Central Asia, but has no known sires or breeds of origin. It was most famously used for the conquering of Europe and Asia, led by Genghis Khan. The Mongolian horse is now used by some Mongols in races as far as 30 kilometers, or 18.64 miles.
Well, folks, you heard it here. Racehorses have a core set of genes that have helped them excel in multiple different ways in the racing industry. These genes affect neurobiology, muscle, and even metabolism within the muscle and the body. Thank you so much to my 3 special guests, and thank you for joining. See you again, Channel 2 News.